Hello, this is Juman at the College of Computer Science and Information Technology at Chakra University.
In this video, I'll explain how to use a BLAST tool and analyze your gene sequence.
First thing is the NCBI. What is NCBI? It's the National Center for Biotechnology Information, and it's part of the United States National Library of Medicine, a branch of the National Institutes of Health. The NCBI houses a series of databases related to biotechnology and biomedicine, and is an important resource for bioinformatic tools and services that might help anyone. It receives data from researchers, develops software for searching and analysis, provides a web access point for the data and software. Lastly and most importantly, it's the gate that will lead us to this video's tool, BLAST.
BLAST is an abbreviation for Basic Local Alignment Search Tool, and it is a program for sequence similarity searching developed at NCBI. It finds regions of similarity between sequences. The program compares nucleotide or protein sequences and calculates the significance of matches. The NCBI Web BLAST offers 5 main search types: BLAST-N, BLAST-P, BLAST-X, T-BLAST-N, T-BLAST-X. In this video, we will focus on BLAST-P. You can access BLAST by the NCBI, and the direct link of a BLAST will be in the description section down below the video. You can obtain the query sequence by several ways, and I will explain one of them, which is how to get the FASTA format of a sequence. By using the NCBI main page, first choose the database you would like to search in based on your requirement. Here, I've chosen a protein database. Then, insert the name of your protein in the search bar. After you get the result, choose the protein that suits your requirement. Once you've chosen the sequence, click on FASTA. Then, scroll down to get the FASTA format. You can either copy the whole sequence or you can easily download the sequence as a file. After you have saved the sequence in the format you like, go to the BLAST page and choose the preferred program. In this video, we'll use Protein BLAST. In the sequence section, you can either paste the sequence you have copied before or attach the saved file from your device. You can also use the accession number of the sequence. Then you can name your search by typing it in the job title section. For the database, there are several databases you can search with. Choose the best one that fits your requirement. In the organism section, you can choose any organism to compare with your sequence. And for algorithm, leave it as BLAST-P. You can characterize your alignment by changing the algorithm parameters. Leave them as default unless if you are aiming for extensive and detailed search. Lastly, click on BLAST. It will take some time to analyze and get your BLAST and result. After you get the alignment result, this page will show up. It contains information about your query, like the job title, program, and database used, and query ID. Then, you'll find the main sections in the alignment process: the description table, the graphical summary, and the alignment section. Firstly, the description table. It displays the output of the BLAST search in a tabular view. It contains multiple fields. First is the description. It is the text from NCBI describing the sequence. Next is the max score. It is the highest alignment score between the query sequence and the database sequence segment. And for the total score, it is the sum of alignment across all segments from some database sequence that matches the query sequence. After that, there is the query cover. It means what percentage of the search sequence overlaps with the aligned segments. Next, we have the E-value, which measures the quality of the match. Lower E-values indicate higher similarity. Then, there is the percent identity. It is the number that describes how similar the query sequence is to the target sequence, which means how many characters in each sequence are identical. The higher the percent identity is, the more significant the match. You can click on the accession number to get to the protein's information, and if you click on the sequence name on the description attribute, it will navigate you to the alignment of that sequence, which will be discussed later. Note that you can sort your results by clicking on the fields you would like your sequence to be arranged with. Next section is the graphical summary. It is an overview of the database sequences aligned to your query sequence. They are represented in horizontal bars, colored coded by score. Mousing over alignment shows the database sequence title. Clicking on the alignment links to the sequence alignment itself. After that, we have the alignment section. Each sequence of the result has an alignment report. You can get each by clicking on the sequence itself, or you can get them all by clicking on the alignment section. It consists of pairs of sequences compared with your query, in which every letter in one sequence is paired with exactly one letter or a gap in the other. The alignment identity shows the number of identical residues in the query and head sequences with the percentage. Positive shows the number of similar but not necessarily identical residues in the query and head sequences with the percentage. Gaps shows the number of gaps in the query and head sequences with the percentage. These values differ from sequence to another depending on its similarity with your sequence. To save your search and settings, click on the Save Search link, then log into My NCBI using the Sign In or Register link at the upper right. Once you do this, your search strategies should appear in the Save Search Strategies tab. Also, you can download your search by clicking on the Download button here. Now, you've finished the process. You can use the results as much as you can, maybe in your research, assignment, or even for exploring.
And that's the utility in Blast. Hopefully the explanation makes sense. For additional help or resources, check the links below the video. I appreciate your like and thank you for watching. It was Juman, the technology lover.